All right. So today we're very honored to have Professor Eve Madar to join us. It was a really great honor to have her on board. So Professor Eve Madar is the Victor and Beinfield University Professor at Brandeis University. She studies the dynamics of small circuits. She is a past president of the Society for Neuroscience, a member of the American Academy of Arts and Sciences, the National Academies of Science and Medicine, and recipient of numerous honors and awards. She has a distinguished record of mentoring trainees at all levels and has published both scientific papers and numerous opinion pieces relevant to the lives and training of scientists. So with that, I will give the stage to Professor Madar, and you can begin.
Oh, I'd like to thank you all for joining us. And because we have a relatively small audience, if any of you want to either make any points or ask questions or raise anything, you should just either, it's fine for you to interrupt because this material will take us as long as it takes us. And I think these kinds of sessions, as I understood you were looking for, are best when they're interactive. So you can either interrupt by unmuting yourself and just speaking, or you can raise your hand and maybe, hopefully I'll see it. And I'd just like to get started. And I'm gonna get started by showing you a picture of my present lab, where everybody, most of you or many of you realize we work on lobsters and crabs. And so these are the crab imitations of most of my present lab, including undergraduates, graduate students, and postdocs and technicians. And this is one of the recent sunny days where everybody was squinting into the sun for this picture. Anyway, just to give you a sense, I would just like to thank all my previous trainees, present and previous trainees. My present lab is here. And then I have a series of lab alumni. These are people who are mostly still in training somewhere else. And then a large number of people who've gone on to industry and government and to faculty positions. And I would just like to say that I'm as proud of the people in industry and government as those who are in faculty positions, and more of our best people are going on to industry and government as along with those who are staying in academic science. And I think the real secret is for each person to figure out how to best follow their heart and figuring out what kind of future is best for him or her. Now, as I've been doing a lot of thinking about science then and now, partially because I was preparing this and partially for other reasons. And I think some things in science have really stayed the same and some things have changed a lot. But in all cases, for those of us who work on what some of you might call model systems, the trick is to extract general principles of nervous system function from model systems, and that's picking the right questions. And whether you work on using mouse or human or flies or worms or crabs or lampreys or whatever, the real trick is to figure out how to match the question to the strength of the organism, because that's where the new insights come from. And for me, I've spent my entire career working on the stomatogastric nervous system that comes from lobsters and crabs. I've had to be very, very, very mindful and thoughtful about making sure that we were doing something that we could do better than anybody working in mouse or worms or flies, because otherwise we would be paying a very heavy price. And so I've had to be very self-conscious and aware about this. And I think many people working on more consensus systems don't think quite as hard about these issues. And it's something that you guys should all think about. Now, just to give you a sense of my career, I started graduate school in 1969. And in the first part of my career, I did what we could call system identification, neurons, transmitters, connectivity. And that kind of analysis of circuit function has remained. It's been part of the work in my lab all this time. In the early 80s, the late 70s, early 80s, we started working on neuromodulation, and that has continued throughout the rest of my career. Then in the 90s, we started working on mechanisms of activity-dependent regulation of intrinsic excitability that became homeostasis and homeostasis of intrinsic excitability, etc. And then this turned into, in the 2000s, major studies about variability and robustness, and which is mostly what we do today. But notice these are all linked, and today we actually go back and forth between all these problems. Now I've shaded in green here when we started using computational methods, and that started in the late 80s. And so everything from then to now has been a mixture of experimental and computational work. And the reason for that, and I think it's really important to explain why that happened, is I'm a biologist by training, and at a certain point it became clear, and I knew this as a graduate student, that it was very hard to tell a story about interacting neurons. The minute you got to more than 2 or 3 neurons, you had to tell a story that was 2 or 3 neurons at a time. And so it became very clear to me that if one wanted to look at more than a few nonlinear processes, you needed tools that were a little bit better than your own brain, because our brains don't allow us to think very rigorously about more than 2 or 3 nonlinear processes interacting. So that's really why I ended up doing theory. Now I'd like to go back to any questions thus far? Everybody with me? Okay. Now I was in graduate school. I started in the fall of '69. So I started in the lab, Balance Helverson's at UCSD in 1970. And I sometimes joke that my PhD is one whose value has accrued in time, because when I went to UCSD as a graduate student, UCSD was maybe, I don't know, the biology department was maybe 3 or 4 years old. So we were among the very first classes there. And it was an exciting place to be. It was a brand new institution. And there was a lot of really exciting work. Now, when I started, my class was a class of 30 in the biology department. And I should just tell you that when we first arrived, there was a great upheaval because there were 13 women in the class of 30. And everybody was afraid that this was going to be the end of civilization as we knew it. And by that following spring, when people had settled into labs, everyone figured out that there was no problem. And for those of you who don't know this, the reason that happened is because the US draft laws changed. And so a lot of men who would have gone to graduate school either went to medical school, or they entered MD, PhD programs, or they went to Canada, or they did whatever. But the pool of men applying to graduate school decreased enormously, and this allowed women to enrich in PhD programs. And so by 1970, 71 or 72, the life science graduate programs in the US basically were gender neutral. And so it's very important for you to realize that between about 1972 and now, it's been 50 years, that entrance to graduate school was basically 50-50 in this country. And therefore, a lot of the discussions about women in science are ones that leave me with very little sense of humor about, because it's just been so long. But when we started, the class before me had 2 women in a class of 30, we were 13, and that was because it was precisely then that the draft laws changed. Now, we obviously, those of us who made it, we just ignored a lot of the overt and covert sexism around. We just literally put up with crap that people today would never dream of putting up with it. But I'm not sure it really bothered us that much. There must have been some people who were driven out, but when people behaved like total jerks, we would roll our eyes and say he's a jerk and then just avoid them, which underplays. I was talking to a colleague of mine who was in the same generation, and what we don't know is how many people were actually driven out of science by the stuff that used to happen that just made me roll my eyes. So we'll never know how many people were driven out, or how many of us just rolled our eyes and just kept on going. Now, I learned a lot of things in graduate school, but one of the most important lessons I learned was that you can't trust anyone else to make sure you aren't making mistakes, and each scientist has to accept responsibility for his or her own work. Now, I learned this because I was doing work quite different from what anybody else in the lab was doing at the time. And I realized sort of towards a year before I was ready to finish that I knew more about what I was doing than my advisor did, or than anybody else in the lab did. And at first I got very angry at my advisor because I felt like he wasn't going to be able to protect me against making mistakes. And then I said, well, how dare you get angry at him if you think you're ready to get a PhD and you're an independent scientist? It's not his problem, it's your problem. And so after a couple of days of thinking about this, I realized that if I really thought I was ready to have a PhD and be an independent scientist, then it wasn't his problem. It wasn't his job to protect me against making it was my job. And I went through this 2 days of being angry until I finally just decided I would have to live with it and accept it and just trust myself and accept responsibility for my own work. And you couldn't, now that didn't mean I couldn't ask for advice, I couldn't talk to people, I couldn't evaluate other people's opinions, but at the end of the day, it was my problem. And I am actually concerned, and maybe we can talk about this later, that our new emphasis on mentoring means that people have grown used to providing responsibility to others for this. Yes, you have a question or comment?
Yeah, so you mentioned like when during your PhD, it was kind of like a lonely road because like you weren't working on like problems related to other people's work in your lab. And it's kind of a similar situation where I'm at now. So I was just wondering how did you deal with this problem, or like who do you eventually talk to kind of just get the conversation going with other people.
So interesting, you called it a lonely road. I didn't view it as a lonely road. I viewed it as my road. I never felt lonely, but there was nobody else in the lab doing similar work. What I did, starting about halfway through my PhD, what I was doing for my PhD is I was working on trying to discover what the neurotransmitters in the lobster stomatogastric ganglion were. So I read everything I could find. There were some very beautiful papers in the literature. And I actually just picked up the telephone and called one of the leaders in the field about one of the assays he was doing. He invited me up. He was actually up in LA. I got in my car and I drove up to his lab, and he taught me how to do single cell colon acetyltransferase assays. He became a very good friend for many, many years. And he and a number of other people that I contacted along the way became part of my sort of, they just taught me things because I asked them for help. Now, and over the years, in that time, so I learned how to do single cell biochemical assays from Dick McCammon. I learned how to do ionophoresis from Yoshikita Koro at the SOC. I learned how to do different things from different people. Postdoc in my lab, Brian Maloney taught me how to do some electrophysiology, and my advisor taught me how to do some other things. So I just put together the help that I needed, but I took the initiative. And, you know, I just walked around and found the help I needed, and I read, and I figured out what I should be doing by reading. So I think it never crossed my mind to expect my advisor to know everything, but at the end of the day, he was very, very helpful in providing sort of the freedom that allowed me to do what I wanted to do, but I had to take responsibility for doing it. Okay, but this is something I realized yesterday, science then and now. So discovering something entirely new was much easier for a graduate student in 1972 than it is in 2022, because so little was known. And, you know, I used to say that in the 1960s, you could go into almost any part of the nervous system with an electrode and make a recording and see something no one had ever seen before. In the early 70s, it was still possible, and people did, to do things that no one had ever done or had dreamt of doing. And so in a sense, our job was much simpler because you just had to do something well, and it almost didn't matter what it was, because short of just going and repeating something that someone had already published, anything you did that hadn't been done, you were likely to stumble on something that was new, maybe a big new, or maybe a small new, maybe just a little new, but new. And I realized that the reality of being a graduate student in the beginning of a field, as the field was growing and opening up, was very, very different than entering the field today. The field today is much more mature, and a lot of the low-hanging fruit in many areas has been plucked, and it's a lot more difficult for many people to do what whoops, I was just reading the chat, now I have to make that go away. To make discoveries. And so I realized something else yesterday, that when I was a graduate student, I was interested in neurotransmitters in the somatogastric nervous system, and I was interested in discovering them because I had the sense that if I characterized them all, I would see some big organizational principles that people weren't revealing by only studying one transmitter at a time. But be that as it may, when I became interested in invertebrate transmitters, I went to the library and I read everything that had been published about invertebrate neurotransmitters. And in 1973, when I did this, it was possible to do that. I wrote a big term paper about that, and then I ended up co-publishing a review article, in which much of that work was put together. But if I asked one of my students today to do that task, they'd look at me like I was psychotic, because there's just, not 100 papers, but thousands and thousands. And so it would not be possible for someone to process the literature the same way that I did in 1973. And I think this is actually something which is very, very, very hard for people who are entering the field, because you can't get your head around an entire literature with the same confidence that I was able to as a beginning student. And maybe we'll come back to this. I think this is actually one of the core problems that new trainees are facing, that the trainees who enter my lab now are facing. It's harder for them to get their head around the intellectual context of the field. Okay, why is this not moving? Okay. Now, I graduated in 1974 with my PhD, and I decided I was, going to do some postdocs. And I spent 1 year at the University of Oregon and then 3 years in Paris. And I loved Paris, and it was a difficult place to be a single female American, because the French at the time didn't believe in single women, and they didn't believe in Americans. They didn't believe in a lot. But the lab I was in at the École Normale was very supportive. So these are things I realized. There was a myth that all the guys used to say that the postdoc years are the best years of your scientific career. And I would say that my postdoc years were the worst years of my scientific career. And they were the hardest years. And I took a long time for me to figure out why. And this is very relevant to people who are postdocs now, because I think it's only gotten worse. They were the hardest years of my scientific career because the future was uncertain and there was nothing, but that's not the real problem. The problem was that as a graduate student, you have a very large peer group, and you have courses and qualifying exams and teaching, and I was a TA and all this. So not only did you have your work to do, but you had other tasks and you were part of a community. As a postdoc, I was one of 2 people in this giant lab who was relatively young and unmarried. And that was okay. Everyone was friendly and fine, but there was nothing to buffer you against the work going slowly. And I think this is the key to it. In graduate school, if an experiment didn't work, well, I probably had to go and be a TA for a few hours so I could get a break or satisfaction from that. As a postdoc, you're getting paid, albeit not very well, just to do the work. And when the guys were saying this is the best time of your career, what they meant is you had no other obligations. But for me, it meant there was no other way to get any sense of accomplishment or satisfaction. I couldn't go into a class and teach something and feel good that at least I was a good TA or a good X and Y. And I think I suffered a great deal this way. And I think many postdocs in today's world suffer with the same problem, that there's no break. And work always goes slowly. Sometimes it goes better than other times, but it's difficult. And I think that this is the core reason why so many people struggle as a postdoc, that and career anxiety. But there were some myths. I was told that I would never get a job if I did a postdoc abroad. Well, I had 2 very good job offers at the end. And the reason I had 2 very good job offers was not because I had done such brilliant work as a postdoc. I still think it's the weakest part of my whole career. But because I had shown independence by just going off to France. And I think being different and showing independence is very much part of being able to convince somebody that they want you as a colleague. So I think paradoxically it played to my benefit. Now there's this very strong conviction among some people that if you want to get a job, you have to work for a famous person in a big lab. I think I went off to a lab with some outstanding scientists who were pretty well known, but not famous the way others might be today. I think there's an issue we can come back and talk about the advantages of being in a big lab and a famous person versus the disadvantages of being in a big lab with a famous person. And that's, I figured out by the time I was a postdoc, I figured out that if you were in a lab with 20 postdocs, and if 2 or 3 of them were doing really good work, if you were one of the 17, the advisor maybe didn't need what you were doing or not doing, because he or she was getting, in those days, he was getting the benefit of the 2 or 3 people whose work was going well. And so more people ended up in career destroying positions in big labs than in small labs, because the PI of a small lab desperately needs to be able to count on the work of the postdocs. And so there, as I said, advantages and disadvantages there. And then today, we didn't have it, this was not something that even existed in my day. You needed to publish papers, good papers, but there was no concept of high profile papers the same way. I published my first paper in Nature, not because it was a high profile journal, but because I was reading, the kind of work I was reading was published usually with a short report in Nature, followed up with 2 years later with a long version in Journal of Physiology. And so that's what I thought you did. You published your first paper in Nature, and then you followed it up 2 years later in Journal of Physiology. And that's exactly what I did. But it wasn't because Nature was going to be high profile. It's because the English neuropharmacologists I was reading were doing that. So that's what I thought you did. I mean, it was really that simple. And I think this whole issue of the contortions that people turn themselves into for the high profile papers is one of the worst soul destroying traps that postdocs put themselves through today. We can again come back and talk to that. Okay, now I've been told any number of times that being successful in science requires giving up having a normal life more than being successful in other careers. First of all, I don't think it's true. I think beginning lawyers work much longer hours than most postdocs or assistant professors, and they probably have much less flexibility in how they spend their time. In our day, in my day, there was no rhetoric around work-life balance. People just made their own decisions and solved their career family issues in their own ways. One of the reasons I went to work with Jacques Souquille in Paris, first of all, she had done what I thought was the most beautiful work in invertebrate neuropharmacology. And secondly, I was just really curious to see how she managed to combine raising 2 small children. She had 2 small children at the time, and doing this outstanding work. And when I got there, I realized that she had created a way of working that was probably unreproducible. She completely solved her own work-life balance in a way that was remarkable and the essence of who she was. You know, she'd walk up the hill to the lab, she'd start her experiment, she'd leave the chart recorder running and the experiment running, go down the hill, have lunch with her kids, walk back up the hill, finish her experiment. And she'd be in and out of the lab 3 or 4 times during the day with, you know, walking her kids to and from school in between. And it was just very, very interesting to see how she did it and to realize that she had just solved her problem in her way, and it worked really well for her. And it was not going to be a role or a model for how other people would do it. And as I looked around, I realized that everybody had solved this problem in a different way. And I would just like to say to those of you who are combining family obligations and work obligations, you just have to figure it out in a way that makes sense for your situation, because there's no obvious single solution. And I talked to my husband about this today. He was a young assistant professor with 3 small, a young graduate student with 3 small children, and then a young assistant professor with 3 small children. And he just learned to be extremely efficient. And then, and he's had a career that's gone on for 60 years. Yeah, for more than 60 years. And at different times, he's had different kinds of obligations, and he just always just kept on working and fitting in all of those obligations along the way. So I think efficiency and not wasting time is the most important thing that people learn. Okay, now I started my own lab in 1978, and I was assistant professor from 78 to 84. And in thinking about it, I realized that the most important thing for my success was having the right friends and colleagues. And there were two kinds of those people. One, I had a colleague, Ron Calabrese, who was then at Harvard, started as assistant professor in doing very similar work 3 weeks earlier than I started. And so during those first few years, he was a friend, a colleague, and a calibration signal for me. We could just keep going in the same way and touching base and got advice, etc. And Coppola White was 1 year ahead of me at Brandeis, and she became a very good friend. And again, she went through everything 1 year ahead of me. And so having someone who was basically a peer as a way of helping us through. And I think having peer colleagues is extremely important. And these were much more important than any of the senior mentors or whatever, because it was these people who really were there to help you touch base, etc. And I think it's really important that people don't overestimate the value of senior mentors and underestimate the value of just having friends. Okay, now I was really lucky, and I call this complete luck, and I'll be with you in a second. I had the right first students and postdocs. Judith Eisen and Scott Hooper were my first two graduate students, and they are both extraordinary scientists, and they were brilliant. And I was just unbelievably lucky. And Chris Lingle was my first postdoc, and he, again, is brilliant and really, really good. And all three of them have become well-known stars in the field. So there was a hand somewhere. Was it?
Yeah.
Was it Asti? Yeah.
Yeah.
Did you want to say something?
Hi, very nice meeting you, and I'm from Israel, and thank you for that. And you seem very honest, and it's, for me, it's really exciting to hear somebody at your level speaking with that kind of grace and honesty. And I wanted to ask you, what did you mean by touch base? What do you mean by that with your peers? Can you give an example, please?
So I'll tell you. So Ron Calabrese was an assistant professor at Harvard, and I was there at Brandeis. And not only were we having to set up electrophysiology rigs and buying equipment, and so we'd get trade advice back and forth, but sometimes something would happen. Someone would say something to me, and I would say to Ron, "Has this ever happened to you?" And he'd say, "No, I've never heard of that." Or then sometimes I'd say, "Did this ever happen to you?" And he said, "Oh yeah, that happens all the time." So when I asked Ron if something had happened to him, very similar to what had happened to me, and he said yes. At that point, I realized that this was a case of people being mean or uncaring or abusing or whatever, all assistant professors, right, male or female. And that was most of the time. And then every now and then he'd look at me, he'd say, "No, I've never heard of that happening." And I said, "Okay, this now is happening because I'm a female assistant professor, not just an assistant professor." It was extremely important for me to have someone at my career level at a different institution, but who was a very good friend, say yes or no, this happens to me. Because otherwise I might misinterpret. I might say, oh, this is happening to me because I'm a woman. And it had nothing to do with being a woman. It had to do being an assistant professor or an assistant professor working on central pattern generators and vertebral nervous systems. And so that was invaluable to me, to keep me grounded in what was the actual reality that pertained to my situation. So I think that's really, really key, that you have people at your peer group who, that you can sort of say, does this make sense? Just to calibrate yourself. And I think that that was one of the most important things that ever happened. Okay. Now, in 1985, we and Pete Getting, who at the time was probably the leader in our field, we said in 1985 that circuit reconfiguration then meant that the temporal dynamics and modulatory environment construct the functional circuit by tuning functional synaptic strength and modulating neuronal excitability. In 1985, this was a paradigm shift and came from the work that actually got me tenure. But the reason I'm bringing it up now is, first of all, there's still people today who don't know this, and I don't understand how they could not know it. But what I realized, again, thinking about this the other day, is that we published 5 or 6 papers, actually published a few more than that got me tenure, but it was my tenure work with Judith Eisen and then eventually with Scott Hooper that led us to this conclusion. And it really was a paradigm shift. And the likelihood that an assistant professor with a small lab, a couple of graduate students, 1 or 2 postdocs today would be able to do something that really was a paradigm shift is much more difficult for that to happen today. And partly there was just more room. And partly it was at a time when, as I said, whenever you did anything, you could do something new. So, and I was pretty smart. That is to say, I was able to understand it. I spent a lot of time talking with Pete Getting, and it was those conversations, who again was in the same field, that actually helped formulate these ideas. But it really was, at the time, in the small circuit world and probably in circuit world, a real paradigm shift. And I don't know how many paradigm shifts happen today from 4 or 5 years of work in an assistant professor's lab. So I think that's, again, a difference. I don't think it's because I'm smarter than most assistant professors today. I think it's just that the field was newer. And so, as I said, if you did anything really interesting and had your eyes open and noticed and you thought about things, you could end up doing that. Okay. Now, by 1980, we and workers in many other small systems, Crickets and Leech, etc., knew that connectivity diagrams were only a starting point to understanding circuit dynamics, and a connectome was necessary and insufficient. Now, we knew this in 1980. Al Silverson wrote a paper about this in 1980. We all had by then gone through the process of trying to establish a wiring diagram and then realizing that they were all different and they all were just a starting point. And we, I think, all thought it was so obvious that most of us didn't even bother seeing it. And I've said this more often in the last 5 years than I did it in the years between 1980 and 1985. And that's because to us working in small circuits, it was obvious. And it's only been since people have been working on the big connectomes that it's been important to say. And I guess this sort of is dismaying because it tells you how poorly information percolates through fields when the field gets very large. And that's a whole other conversation we can have about the difference between what it is to be part of a mature field as opposed to a beginning field. Okay, now other myths. Most graduate students and postdocs think that running a lab and teaching is harder than being a graduate student or postdoc. It's just not true. It's not true for lots of reasons, but mostly it's not true because most people actually do better when they have tasks that can, again, buffer themselves from one or another thing not going well. And I might remind all of you, if anybody in the audience says, oh, you couldn't do what I do today, well, of course you couldn't do what I do today because you haven't been in the field for 50 years. I think it's really important if you look at your advisor and you say, I couldn't do what they do, to ask, not could I today do what they do today, but could I do what that person did when they were a graduate student or postdoc or an assistant professor. And that's really the appropriate comparison. And trainees often make the mistake of comparing themselves against someone who's 20 years senior. And that's just not a right comparison, because what you really want to know is what was that person like at your career stage, and you don't know that. And so you have to be really careful about making those comparisons. We'll talk about that now. I don't have that much more time, but I want to talk about getting stuck. And everything I ever did that turned into a major discovery came from being stuck. And it's really, really important to realize that when you get stuck and you eventually turn the problem around, that's when you discover something new. Because you get stuck when your preconceptions, the way you thought the world was, turn out not to be right. And so being stuck gave rise to all of our work on circuit reconfiguration in the 1980s. In 1992, to the development of our first homeostatic model for intrinsic excitability. It also drove us to probe the basis of robustness in the face of variability during the past 15 years. And I would say that as painful as it is to be stuck, it's, the image I like to see is you're stuck, you're headed into a corner and you're facing into the corner and you're stuck and there's nowhere to go because you're facing into the corner. And all you have to do is turn around and face back into the room, back out into the world, and look at it with new eyes, and you'll see the answer, because the answer is there, but sometimes the blinders of being driven into the corner hide it. And I think it's really, really important to try and do experiments. And then when you get stuck, and once you figure out that, it's not that you've done the experiment wrong, but you're stuck because there's something you don't understand, that you just turn it around and look back out and then say, well, what is my animal telling me? What is the world telling me? And as I said, I think everything important we ever did came from being stuck. And the important thing is not to get flipped out about being stuck, but to really figure out why you're stuck, what the essential problem is, and then say that problem is the answer somewhere. Now, the last thing that we really want to talk about is being wrong is the worst thing that can happen to you as a scientist. And as a young woman, I was taught this was really bad to be wrong. And I thought a lot about being wrong, because being wrong is awful. One hates being wrong. And obviously, if you're wrong and you publish something that's wrong, you have to fix it. I mean, you just have to figure out how to fix it. If it's a tiny little detail, you can fix it in a little way. If it's a really major thing that you made a really honest, then you fix it, even if you have to retract paper, a correction, whatever. But, and this is not a justification for being wrong, you should always try and do all the controls that you can do to try and not be wrong. But the only way to avoid being wrong ever is to repeat what is already known. And this is really, really, really important, because if you're really trying to work at the boundary between what is known and what is not known, you are bound every now and then to make a mistake, because you won't necessarily know how to interpret something because there's something that isn't known or understood, or you don't do the right controls because you wouldn't even know what the right controls are. And so, if you're honestly trying to push the boundaries of new knowledge, you will every now and then end up making a mistake. And you have to deal with it as honestly and as openly, as transparently as you can. But you can't be so fearful of making a mistake that you just repeat what's already known. I mean, if you go into an organic chemistry lab and repeat the lab exercises that have been done millions of times by students all around the world, you should be able to get the right answer, right? Because it's known what the right answer is. But if you don't know what the right answer is, you have to do, to the best of your knowledge and ability, the right controls and the right experiments, and analyze the data correctly, and every now and then, and the right statistics, and every now and then you'll get something wrong. And often there's information in the way that's wrong, but you have an obligation to fix it once you figure out if it's wrong. But it's also often the path to new knowledge. And so I think one should always try and do something new and different, knowing that every now and then this is going to happen. Does that make sense? Now, I should probably stop pretty soon, but I'd just like to say that Eric Kandel was sitting at we were standing at a poster session in 1976. And Eric turned to me after everybody left, and he said, "You want to know what the secret of success is?" And I said, "Sure." Eric wasn't so well known then, but he still was a senior person in the field. And I was a postdoc. And he looked at me, he said, "Just keep working." And I think there's some really, really, that's deep wisdom. If you stop working, you won't make findings. And if you keep working and you keep working and you keep working, you will find something interesting if you have your eyes open. And I think what I could do is I could stop here and just open things up for any other questions and comments. I mean, there's more stuff we could talk about, but this is as good a time to stop as any. Any other questions or comments? Thoughts?
I think so if we could start the questions, maybe I just want to thank Professor Marder first, and then we can go into questions, and I will stop the recording. So I have to say this is really, we're very lucky to have you on board, and hopefully this has been a really rewarding conversation for everybody. And we just want to thank Professor Marder for the time and effort you put in this. And for that, I will stop the recording, and everybody can start asking questions.